Okay, great. Yay, people came back. That's always promising. Welcome back to Basic Plant Science Lab. Let's see here. What do I need to do? I need to share this. And we'll start with just sharing the PowerPoint slide. And then, if need be, I will, oops, whoa, not meaning to jump into it that fast. I'll start with the PowerPoint slide, and if need be, I'll pull up the lab manual as well. Let me just, sorry, I'm multitasking here. Okay. Cool. So you guys had lecture, so some of you may not be enrolled in lecture because these are technically two separate classes, but some of you may be enrolled in lecture, and so I guess you guys probably had your first lecture yesterday with Dr. Tripathi. Yeah, great. Yeah, so like I said before, the lab is self-contained, so if you're not in the lecture, that's fine. But I've often heard in the past that if you are in the lecture, being in the lab can be very helpful because there is a lot of overlap when it comes to material. So that's a good thing. Okay, so let's jump into it. It's already 6:02, but I always say as soon as we get started, the sooner we get finished. So again, if this is your first time joining us, I'm Dr. Rebecca Panko. This is my email address, so feel free to email me if you have any questions at any time. And last class, so you guys would have done the first assignment. So how did that go? Did everyone have an okay go with that? You can write in the chat box or you can just speak out. Any problems? I did see some of the grades already. Several people are getting hundreds. Great. Okay, so people are saying it was pretty straightforward. It was fun to do it. Well, that's good. It's a nice when an assignment is fun. But now you guys see they're not meant to be scary assignments. They're just meant to help you digest the information slowly over time as versus trying to cram. So hopefully now you guys are less nervous about them. There will be another assignment available today that will be due by Saturday. If you didn't do as well as you were hoping, not to worry. Now you kind of have a feel for what they might look like. As I said during the first class, my best advice is to obviously either join me here for the live classes, or if you are busy during this time, watch the recording before attempting doing any of the assignment. Read over the lab manual before attempting the assignment. Remember, the PowerPoint slides are always going to be available to you for your reference as well. But I do recommend that you take notes when you're watching or when you're watching the recording or when you're here with me now. I do recommend you take notes. That way you don't have to re-watch the recording if you've already watched it the first time, right? Okay. So last time we reviewed the syllabus, which is still available on Blackboard, talked about kind of the importance of plants, and we did a little virtual tour of plants around campus, right? And we came into contact with some new terms likely for you guys, including things like monoecious and dioecious. We also talked about things like evergreen and deciduous and things like that, right? Conifers, which are part of the gymnosperms, angiosperms, which are the flowering plants. So we actually covered a lot of ground last class. I don't think today's class is going to take as long, so let's just jump into it. What are we going to learn about today? Today, we're going to learn about the stem. So it's maybe not the most exciting part of the plant, but it's a pretty important part of plants. We're going to talk, before we can talk about the stem, we kind of have to talk about what makes up a plant, rather than just focusing only on the stem. We kind of have to figure out what other parts of the plant there are. We're going to talk about something that's called primary versus secondary growth. We're going to compare dicot stems and monocot stems. Don't get worried about those terms. You will learn about them today. And then some examples of stems that have certain special adaptations that allow them to do cool things. Okay. So before we learn about the stem, we have to look at the plant body in general. So this is an example of a plant, a representative plant. And the plant body, here, let me, on my other thing here, I just want to see where I'm going with this. Yeah, the plant body consists of a few main parts, okay? So don't get bogged down on all the names that are here. Since today we're only really going to focus on the stem, but the plant body, here, hold on, has these general parts. So there's a root system, right? Plant roots, and their main job is to anchor or keep the plant in the ground so it doesn't fall over, but then also to absorb water and minerals. So that's the main job of the root system. Then you have a shoot system, which is the stems and leaves, and then also the flowers later. So the stems are the pokey, sticky type parts of a plant, right? And the job of the stem is to elevate the leaves, because the leaves need to be out so that they can get sun. And so the job of the stem is to allow lots of leaves to be on one plant and have them elevated so that they can face the sun. And then I said flowers are also attached to stems, but we'll learn about that a little later. Then the other part, so you have the root system, which is underground usually. You have the shoot system, which is above ground. And then inside of both of those, you have this vascular system. So we're humans, right? And we have a vascular system as well. We have arteries and we have veins, and those tissues allow blood and things to move around through our body, right? That's our vascular system as a human. Plants also have a vascular system. However, instead of veins and arteries, they have xylem tissue and phloem tissue. Xylem tissue, the job of xylem tissue is to move water and minerals from the root to the rest of the plant, right? So from the roots to the leaves. That's the job of xylem tissue. The job of phloem, so you say xylem, like xylophone, that same xylem tissue, and then phloem tissue, so it's a pH, like phloem tissue is the other one in the vascular system. And phloem tissue conducts or transports food, so the products of photosynthesis, which we talked a little bit about before in the first class, but basically sugar, right? So xylem tissue transports water and minerals from the roots to the leaves. Phloem tissue conducts sugars from the leaves, where photosynthesis occurs, down through the plant body all the way down to the roots. Okay, so now we know the parts of the plant body. So part of the reason I'm going over some of these terms is because they are in your lab manual. So I have the lab manual up here. Just as a reminder here, let me do this really quick. I'm going to stop sharing this slide for a second so that I can share my screen, just to remind you guys, right? So this is the Blackboard page for the lab course. If you go to course documents, have lab manual there, right? And so the stuff that we're covering today, that's the first lab there, is stuff here, right? So look, I just talked about the plant body is divided into the different parts, right? Shoot system and root system, etc. So this is where I'm getting the information that I'm sharing with you guys today. Oops, that's not what I want to push. There we go. Okay. So, talking about the stem. So some of the things that are on the stem, right, are, let me just look here to make sure, right, so we talked about the xylem and the phloem. Not going to really go into leaf today because we're going to go into leaf next class. Right, But there are parts of a stem. So this is an example of a stem. And what I want to draw your attention to on this slide is, sorry, I have too many screens. Right. What I want to draw your attention to on this slide, let me annotate because I'm super awesome at annotating. It's like I'm the best in the world, are these terms. So the apical bud and the axillary buds. Okay? Apical or terminal bud and axillary buds. What those are areas of growth. So maybe you've heard of the term stem cells before, right? Maybe that's something you've heard of before. And a stem cell is a type of cell that can basically divide and turn into all different types of tissues. So these buds are similar in that they have special cells inside of them that allow them to grow the different tissues needed for the shoot system. So for example, this terminal bud right here, this is going to divide and allow new stem tissue to be grown. But sometimes when it divides, it'll actually allow, that's supposed to be a leaf, new leaf tissues to be created. So the terminal bud is involved in making the plant longer, right? Lengthening the plant. And then you have these axillary buds. So this area between, so this is a leaf, right? You can see all of these are leaves. The area between the leaf and the stem, that region, we call an axil. And that is where those axillary buds are. So there would be axillary buds here. There would be axillary buds here. Buds that are located, so buds, right, that are located in the axils of the leaf. So this region right between the leaf, where the leaf meets the stem. And what those will allow the stem to do is to branch. So eventually this might make a branch, right, that's supposed to be a leaf. I'm so great at drawing. This can make another stem tissue to make a branch. So the terminal bud or the apical bud is responsible for making the plant longer. And then the axillary buds are responsible for branching. Okay. Let's move on. Okay. So that's kind of going to start to lead us into this idea of primary versus secondary growth. So we're going to talk about these things, so bear with me because there's going to be some terminology here, but it's terminology that we're going to see in many, many, many labs. So don't get too nervous today if you're seeing a lot of new terms. Just like the last time when you first saw the word monoecious and dioecious, you were like, oh my God, I am in the wrong place. But then I say monoecious and dioecious over and over and over again, and it's like, okay, I can handle this, right? So it's going to be the same thing. Don't get nervous when you see new terms. All right. So primary growth. Primary growth is responsible for making the plant taller, right? So lengthening. So here I have primary growth in red, right? So see this little tree here? Look at this little tree. It's little, needs to get taller, right? Needs to get taller. Oh, I have a question here. What page can I find the intro to the plant? What page on the lab manual? On the lab manual, they kind of jump right into, they don't do a great job of kind of introducing you to the plant. I guess the first page of the lab manual would be your best bet because it talks about the parts of the plant body. It talks about the stems. It talks about the leaves, and then jumps right into the terminal bud and things. I added a little more content here to make it more clear because the lab is very sparse when it comes to talking about this. Yeah. So the first page would be your best bet. Okay, so look at this little tree, right? Super, super small. First thing it needs to do is it needs to get taller. And the way it's going to do that is via primary growth. So by that apical meristem, right? So it's going to get taller via primary growth. However, eventually the tree is going to be pretty tall. And you can't, so if you're a plant and you want to get super, super, super tall, you can't get super, super, super tall without getting wider. Because if you're like a little pencil and you want to get to be 90 feet tall, No, that is not going to work. You need girth. You need to like get thicker in order to get taller. And so that is because of secondary growth. So you see here in this older tree, right? Secondary growth is the growth that allows the plant to increase in width of the plant body. So primary growth, length, secondary growth, width. Get wider, right? Secondary growth. Okay. Oops. So let's talk a little bit about these two types of growth. All plants have primary growth. So I have a question here. Does the growth ever turn stagnant? I don't really know what you mean by that. Do you mean, do the plants ever stop growing? Do the plants ever stop growing? No. The short answer to that is no. If the plant is alive, it is growing in some way. There might be regions on the plant that are no longer growing in a very old plant. But if the plant is alive, it is still growing in some way. That's the easiest way to answer that without getting too complicated. All right. So primary growth versus secondary growth. All plants have primary growth. I'll repeat that. All. Every single plant. All plants. So if I ever ask you a question about if a plant has primary growth, it has to, right? Every plant has primary growth. Every single one. But not all plants ever go through the process of secondary growth, right? For example, without even showing you slides, I can say, think about a grass, right? Think about a grass. A grass only needs to get, whatever, this big, right? It's pretty much made of leaves. And so a grass is never going to need to get thick and wide, right? Because a grass doesn't need to be 90 feet tall, right? So a grass is never going to undergo secondary growth. So there's some plants don't have secondary growth. All plants have primary growth. Only some plants have secondary growth. Okay. So I've color-coded these following slides to make them maybe a bit more digestible. This is material directly from the lab manual where they just throw terms at you. So I'm going to try to walk you through it. And again, I've color-coded these slides. So you see, for example, primary growth, I have it in red. And the primary meristems I have in purple, right? Okay, so let's read this. So this is talking about herbaceous stems, right? Maybe like a grass or something like that. So primary growth results from the apical meristems, right? That's what we talked about before. Here, I'll use the same color as I used before for primary growth, red. So primary growth comes from those apical meristems. That's what I already talked about. That's that terminal bud, right? Apical or terminal bud. That's what the region that you're going to have primary growth occur at, because it's the region that's responsible for making the plant taller, right? Lengthening. And this apical meristem, right, or terminal, so this is in that terminal bud or apical bud, there is this thing called a meristem. And a meristem is a type of tissue that is very similar to like a stem cells. Meristems they're cells. Meristems are made up of cells, and those cells can divide and grow into all different things, right? They can divide and grow into a leaf. They can divide and grow into parts of the stem. Down in the root, they can divide and grow into more root tissue. So at that terminal bud, there's these special cells, call them meristems. And the first thing that the meristem in the apical bud is going to do, it's going to divide to give rise to what we call the primary meristems. Remember, this is going to be color-coded, so let's go through it. So this is a zoom in. We just zoomed in to the top of that terminal bud, right? That apical bud, same thing. Terminal bud, apical bud, same thing. And we're looking at that apical meristem. It's big here, so let's look. So look here, apical meristem, right? It's located right in the middle. And that apical meristem is actively dividing to create new tissues of the plant, because you can't you can't just go from like nothing to something. You have to go through these processes to grow. You could think about it even like with a human, right? If you have a baby and the baby is really, really small, and it's, its fingers and toes are really cute and small, over time, right, that baby needs to grow into a grown-up adult, right? And it's not going to be like one day it's a baby and then one day whammo adult, although it might feel like that sometimes, right? But no, it's like it takes time, right? It's a slow process. The little cute fingers don't go from being nice little cute fingers to whoa, big old hands, right? This is a process. So similar process is happening here. That's the point. So here's that apical meristem, and that apical meristem is going to divide and create these primary meristems. So let's look at them. So there's 3 primary meristems. They are called the protoderm. Proto means beginning of. Derm means skin, like dermis, like epidermis, right? So the beginning of skin is the protoderm. The procambium, which is the beginning of cambium. We'll talk about cambium. But I'm just trying to get you comfortable with these big, scary words. And then the ground meristem. So at the tip of a plant, there's a terminal or apical bud. Inside of that terminal or apical bud, there are special cells called meristems, the apical meristem. Apical meristem is going to divide, and the first thing it creates is these 3 primary meristems: protoderm, procambium, ground meristem. And then these primary meristems, right, protoderm, procambium, and ground meristem, are going to actually make the tissues of the stems. So let's look at that. So as I mentioned, the word protoderm means beginning of derm, right? So protoderm leads to skin, basically, skin of the plant. So the protoderm is going to make the mature tissue epidermis. So plants have epidermis just like us. Just like us. It's not the same, right? It's plant epidermis, but they have epidermis just like we do. So protoderm makes the epidermis. The procambium makes xylem and phloem. And then the ground meristem makes what are called the cortex and the pith. Those are names of tissues inside of the stem. So right now we're talking about pretty much the stem, right? So the protoderm is going to create the epidermis of the stem. The procambium is going to create the xylem and phloem in the stem. And then the ground meristem is going to create the cortex and pith of the stem, all right? So we're going to look at that. We're going to look at how that process is. Are there any questions at this point? I know it's a ton of new words and you're like, no, but it's going to be okay. Like I said, we're going to have a lot of repetition. What's the cortex and the pith? Good question. So we'll see on the subsequent slides that they're part of the stem. And what they really are you have the epidermis on the outside, and you have the xylem and phloem conducting tissues on the middle, and the cortex and pith are kind of like surrounding them. We'll see on these slides. So I just told you how it happened, but let's look at it with our eyes in these different schematics, because I'm kind of a visual learner. Maybe you are too. And so it's one thing to read that the protoderm makes epidermis, okay, but it's another thing to see what I'm talking about. So here is a cross section through a stem, right? I had a stem, I cut it in half. Now I'm looking at the cross section. This would be a cross section of a stem where the apical meristem, remember that first meristem that I talked about, has already divided into the 3 primary meristems, right? And those 3 primary meristems are, as I already said, your ground meristem, which is going to create your cortex and pith, your procambium, which is the meristem that's going to make your xylem and phloem, and then your protoderm, which is the meristem that's going to make the epidermis, right? Okay. So now these meristems are ready to go, and they're going to make these tissues in the plant, in the stem. So let's see. Okay. So look at that. So procambium was this little, here, I'll get a purpley purpley. Where are you? Here we are, purpley. Procambium was right here, right? These little, looks like a seed almost, right? Procambium is these little guys here. And what do you know? Look down here. So the Procambium, where it's still showing it to you, is in the middle of that thing, and the xylem and phloem are being created next to it, right? Because that's what the Procambium is dividing into. The ground meristem, you see here, it's pointing to the middle and also the outside of the stem cross section. And what do you know? Here you go. You have pith and cortex. So that's made by that ground meristem. Don't worry about pith, Ray. Don't worry about that. And then, as I said, protoderm, right? It's going to grow into that epidermis, and there you go, right on the outside where you would expect the skin to be, right? The epidermis. So just because I like color coding, remember purple is the primary meristems and red is the primary growth tissues. So in order from the top, ground meristem is going to make, boom, pith and cortex. Procambium, the second one, is going to make, boom, primary phloem, primary xylem. And then protoderm, which is on the bottom, is going to make, boom, epidermis, right? Okay. So now we know a little bit more about how stems grow those tissues when they're making the primary growth tissue, right? Now we know a little bit more about that. So let's talk about this in dicots and monocots. So we didn't talk about dicots and monocots last time, but basically dicots and monocots are parts of the flowering plants. So dicots and monocots are parts of the angiosperms. And when we get to the angiosperm lab toward the end of the semester, we're going to go into this in real detail. But right now we're talking about stems, and so it's important to kind of compare these two because most of the stuff we talk about when we talk about plant anatomy has to do with the angiosperms. So let's look at these dicots and monocots. Dicots, and sometimes, I don't know in your lecture, if you're in the lecture portion or in the book, it might say eudicots. Don't worry about that for me. For me, I'm just good. I'm cool with dicots. If you, either one is fine, but dicots is what I'm used to, and I'm fine with that. So dicots are the larger group in the angiosperms. They have the most species. They have about 200,000 species. So remember, the plant kingdom only has about 400,000 species in them. And over 90% of those are in the angiosperms. And the majority of those are these dicots. So dicots are the largest group in the plant kingdom. And they're super diverse. So most of the plants that you see on the daily are probably a dicot, because dicots, like I said, are the most common or most prevalent. So you can have trees, you can have shrubs, you can have herbs, etc. in the dicots. There's a few examples here. Even the cactus is in the dicots. Okay. And then you have the monocots. So if you're from the Caribbean or even South America and even parts of Micronesia and Asia, where it's a bit more tropical, you may be surrounded more by monocots. Monocots tend to be in those kind of habitats. And they are still pretty big group. There's about 90,000 species, but like half as many as the dicots. And some of the familiar plants that you might know about that happen to be monocots are grasses, lilies, orchids, cattails, which are a type of grass, palm trees, bananas, and rice. So those are all some examples of monocots. And you're like, why is Rebecca telling us about this? Because there are differences. So there are differences inside of these plants. So the dicots and monocots have some slight differences when it comes to the stem. That's why I'm mentioning them. So let's look at dicot first. So normally what I would have done in lab is we would have had this plant, coleus, and we would have kind of like chopped off the tip of the plant to look at under the microscope. And we also have prepared microscope slides, which I'm going to show you now to kind of walk you through some of the stuff. It's okay. Actually, the slides that are included here are the slides you would have. So fair warning, not fair warning, but I guess spoiler alert. So normally we would have taken the top of the the plant stem and made a microscope slide with it. But normally the microscope slide that we make is not good. It's not usually one that you could see a lot of stuff on it. So we end up using the prepared slides, and I have those here for you. So we're going to look at the prepared slides just like you would have under the microscope in the lab. So it's the same exact slides. Okay. So here's a tip of the coleus. So this should look familiar, right? This looks a lot like this, right? Sorry, didn't mean to yell. This looks a lot like this, right? The thing on the right. And that's because that's what it is. So here we go. This is the tippy tip of that of a dicot in this case. And so some things are going to pop up, but don't worry. So look, so these are stuff we've already talked about before. Let me annotate with, and I'll stick with purple because that's what I was using for those meristems. So here is that apical meristem again, right? Right at the tippy top of the plant. And remember that that apical meristem is going to divide into the 3 primary meristems, right? Procambium, ground meristem, and protoderm, right? So this is where those are located. And the reason I'm showing you this is because, right, so as a prompt, this could be helpful for you when you guys are doing some of the assignments and whatnot, is to quiz yourself, right? So we just talked about it, but what mature stem tissues does the Procambium make? So a hint is that look where it's located, right in the center of the stem. And what, let's look here. The Procambium is going to make tissue that's going to grow like this. So that looks like a tissue. I should actually make that red. So what tissue is the Procambium going to make? Yeah, so somebody wrote xylem and phloem. Yes, right? Look, so this is the Procambium is going to make the xylem and phloem. And remember, the xylem and phloem, and it ultimately is going to go all the way down to the roots. If I can draw, right? And all the way up through the leaves. So yeah, so the Procambium makes xylem and phloem. Next, we have ground meristem. Remember, ground meristem makes 2 different tissue types, and they're not on the inside and they're not on the outside. They're in between that. So what are the 2 tissue types that the ground meristem makes? It's not xylem and phloem. Cortex and pith. Exactly, right? Cortex and pith. Very good. And then, oh my gosh, come on. And you can tell where this is going. Now we're at protoderm, right? Remember protoderm, beginning of what? Beginning of derm. What is derm? Derm is skin. So what is the protoderm going to be making right here on the outside of the plant, right? Epidermis. Exactly. See, you guys are scientists. You didn't even know it. Good job. Good job. Okay, cool. What am I saying with node and internode? Don't worry about node and internode right now. Don't worry about that right now. They're just regions. They're just regions of the stem, but don't worry about them now. All right, so now I'm going to show you a video. Depending on your bandwidth, the sound might not work. I'm going to put the captions on. But so some of you may hear the sound and some of you may not hear the sound depending on your bandwidth. If you can always pull up this video on your own. Basically right now we're going to act as if we're going to make a wet mount of the stem tip. Instead of looking at the stem with the apical meristem on the top, we're going to do a cross section of a stem. So let's try this, see if this works. I think this should work. Okay. I have sound. Let me put captions here. Let me pause. I hope there's not an ad. Sometimes there's an ad. No. Okay, good. Hold on. Wait, hold on. Hold on there. All right, so let's put captions. Okay. And I'm going to make it big. Can you guys see the YouTube? I lost the chat box. You guys can see the YouTube? Okay, cool. Good. All right, so hopefully some of you hear it. Maybe some of you won't hear it. I'm going to put the captions on. But you could always watch the thingy later. Okay, so I'm going to play it.
Sections of plant tissue by hand can be easy if you know how. Let me show you. Always begin with your slide tables closely. So for this demonstration of hand sectioning, I'm going to use foliage plant.
Same plant we used just now.
Harvest the stem sections. So you always want to begin by having slides way.
That's just water.
To make the hand sections, you want to use a fresh blade. Plant material moistens and when the blade loosens. Then to make the cross section, hold your material firmly in your hand and you want to draw the blade sections and make multiple sections. Some but just right. So as you draw it through, now, this one just want to leave it unsafe. For this, want to stay
Okay, so that I like that video because it kind of gives it shows you how easy it shows you how easy it really is to look at plants under the microscope, right? I know, like, if you've never done it before, you might think like, whoa, like, Rebecca's showing us these slides of the plants, and oh gosh, I can't even imagine. And it's really not as, science is not as difficult as it makes sometimes we think it is, right? Sometimes it's literally just taking a razor blade, cutting a piece of stem, putting it in a drop of water, and putting a piece of glass on top and putting it under the microscope. So I'm just trying to demystify it a little bit, right? So you saw what her slides look like, right? And they're okay, they're pretty good, but they ain't great, right? And that's often what happens when we're in the lab and we're trying to make the slides too, because it takes a lot of practice to be able to get the thin, awesome thin section. I'm horrible at it, but I've had some students over the years that first time I was just like, wow, you should do this forever. Okay, so let's look at some of the prepared slides to see. So now we're looking at a cross section through a dicot stem. We just made the wet mount, right? I'm going to look through a cross section of it under the microscope. So let's move this. This says Helianthus stem, which is a sunflower stem, but don't worry, the Coleus stem looks the same. So here, what I've done for the slides to help you guys is I have unlabeled and then I have labeled so that you can pull up the slides that are unlabeled to kind of try to quiz yourself when you're trying to study stuff. So here's our cross section through a stem. Let's look at some of these tissues that we talked about before. So what do I want to focus on? I want to focus on the ones that we've already talked about. So I'm going to use red because that's our primary growth tissue color. So here's the epidermis, right? A single layer of cells right on the very outside of the stem, right? Let's see. Here is our xylem tissue, and here is our phloem tissue. And so that procambium that we talked about before in the previous slides, it would have been located right here, right? See where I'm drawing with the purple? Would have been located right there. And as it divided to make the xylem tissue and the phloem tissue, it makes the xylem tissue toward the inside of the stem, right? This is the center of the stem here. And phloem tissue to the outside of the stem for the primary phloem. Okay. So we have the epidermis, which we already covered. Here's the epidermis again, since I accidentally erased it, right? Epidermis. Here's our xylem and our phloem. And then the pith. So the ground meristem made those two mature tissues, right? The pith and the cortex. The pith is this whole area in the middle. This whole area in the middle, pith region. So all of this is pith region. And then the cortex is kind of like the other part that's kind of around the other. So in between the epidermis and the pith region, that's all your cortex. So like this is all cortex. This is all cortex. This is all cortex. And likewise, I'll use black since it's already circled in black here. So as I pointed out, this is remember your primary xylem and your primary phloem, right? And they're located in what are called a vascular bundle. So here is one vascular bundle, right? Here's another one that's already circled for you, vascular bundle. Here's another one, right? Here's another one. And each of these vascular bundles has inside of it xylem and phloem. So each of these. So they go all the way around the stem in a ring. Okay. Now we're gonna do a close up here. So we were here. Here's our xylem and phloem tissue, and we're going to kind of zoom into a higher magnification to look at it. So here is our vascular bundle again, right? Here's our vascular bundle again. And inside of that, oh, I have to get out of annotate to move forward. That's right. So don't worry about this for now. Don't worry about this. But I'm just showing you close up, right? This is the xylem tissue of the vascular bundle, and this here is the phloem tissue of the vascular bundle. So what is moving through this xylem tissue? What's moving through that? liquid gold, cheese whiz, beer. What's moving through that xylem tissue? Ketchup, chlorophyll, not quite. So remember, we talked about how in humans we have a vasculature, right? We have arteries and veins that move blood and other products, right? Yes. So let's see. Yes, water. So we have a few students here saying water. Exactly. So water is moving through the xylem tissue, right? From the roots to the leaves. Agua, yes. And what is moving through the phloem tissue, right? Relish, pasta sauce. Water's moving through xylem. What's moving through the phloem? Sugars. Yes, sugars is moving through the phloem. Okay, good. All right. I know. Stems are not very exciting. I'm trying, guys. I'm trying. Okay. And then this is just a close-up of the phloem. So we were here looking at the primary phloem. Let's a close-up of the phloem. And I just later on you might cover these terms in lecture if you're enrolled in the lecture course. So I'm just showing you where they are, but these are the cells. These are the cells that make up the phloem. Don't worry about those too much for right now. Okay. So that was a cross-section through a dicot stem, right? Cross-section through a dicot stem. Here, I'll go to the unlabeled one, where you have the pith region in the middle, the cortex between the epidermis and the pith region, epidermis on the outside, and then these vascular bundles that go around the outside of the dicot stem in a ring, right? We can't see the whole thing, but trust me, this whole thing is going to keep continuing this way, and this whole thing is going to continue that way around the outside of the dicot stem. So this dicot stem. Now let's look at monocot stem. Oh my gosh. Go, go, go. Okay, yeah. Now let's look at a cross-section through monocot stem. So different. Looks super different, right? So let's look at what we're looking at here. What are we even looking at here? So there's some familiar players in that here, I'll use red. Use red. So you still have an epidermis right on the outside. Ooh, look how good. Ooh, wow. Whoa. Got excited. Sorry. Epidermis is still on the outside, right? Still somewhat of a pith region here in the middle, but it's a little less clear because it's not like a big empty area anymore. There's all these little weird faces all surrounded throughout the plant. But there still is, like, you could still consider this like cortex region and maybe this pith region, but it's a little less clear than in the dicot stem. And then the other thing I want to point out is these little things that to me always look like faces. But so each of these things all spread out are the vascular bundles, actually. So if we go to the next slide here, don't worry about sclerenchyma right now. Don't worry about that here. Get rid of that. Don't worry about that. But look, so here, remember I said each of these is a vascular bundle. And just like in the dicot stems, the vascular bundles are going to have xylem, which you guys just told me moves water, and phloem, which you guys just told me moves sugar. And this is the same thing here. Here, the larger cells, the ones that kind of look like eyes, are the xylem tissue. And then the kind of bluish, greenish color is the phloem tissue of the vascular bundle in the monocot stem. So thinking about our dicot stem that we just looked at, and now our monocot stem that we just looked at, what do you notice? So if you were to compare the two together, right? How do the tissues in the monocot stem differ from the tissues in the dicot stem? And so I'll give you a hint. It has to do with the vascular bundles. So how are the vascular bundles arranged in a dicot stem? We can go back. How are they arranged? Uniform, yes. Uniform, sure, is a good way to say it. But what about, where are they? Where are these vascular bundles in a dicot stem? They are arranged in a uniform fashion, for sure. But where are they located? And how are they uniformly arranged, right? So kind of like this. They are concentrated near the epidermis, yes. And what shape do they make inside? So all the vascular bundles make what kind of a shape? It's like this. A donut. Yeah, it's true. But I love that. Yes, it is a donut shape. But what we say is that they're arranged in a ring, So the vascular bundles and here, I'll make this more clear in the comparison slide coming up. I like donut though. I think I might have to change it to donut. That's good. I like that better. That's more fun. Yeah, so the vascular bundles in the dicot stem were arranged around the outside in a ring. And the vascular bundles here in the monocot stem are not, right? They're scattered. They're scattered throughout. So let's look at them side by side. Maybe a little easier to see this. So this is what I just said, but it's on a slide now. Yeah, so in the dicot stem, which is on the left, right? Here, I'll draw this. Dicot stem on the left. Here's the vascular bundle. Here's another one next to it, right? And they're arranged. Oops, nope, don't do that. They're arranged in a ring or donut, right? Ring or a donut. Whereas here, all of these little dots are the vascular bundles in a monocot stem, and they're not arranged in a ring, right? They're all scattered. So that's one thing to keep. That's one thing to make a note of, is that you can easily tell monocots and dicot stems apart because of the way that the vascular bundles are arranged. Okay. Let's go forward. Okay, so now we have to talk a little bit more. And I think there was a question that I missed before we watched the video. So is there anything special about dicot anatomy that we should focus on? Yes. So in the case of the stems, the main thing to focus on between the dicots and the monocots is that vascular bundle arrangement. Yep. Yeah, I thought maybe I missed a question back there. Yeah, sorry. I guess I did. I have a question here. Because of climate, what do you mean, Diane, in terms of because of climate? And feel free to unmute if that's easier than typing if you want.
Oh, because you said that. Can you hear me?
Mm-hmm.
Can you hear me? My connection's kind of okay. Because you said monocots are usually found in the tropics or in warmer climates.
Mm-hmm.
So does it have to do with like water absorption?
That's a very great question. So as we move through this primary growth versus secondary growth, we're kind of going to touch on that. But the short version is it doesn't necessarily have to do with climate as much as it has to do with who does secondary growth. So in general, dicots do secondary growth a lot more than monocots. And so the arrangement of the vascular bundles in the dicots makes it easier to make that transition into secondary growth. And I think as we go through these next slides, it might be a bit more clear. But that's a really good way to be thinking. It's like, well, monocots live in the tropics, so does that have to do with the vascular bundles? But no, it's more that they tend to only do the primary growth. Good question, though. All right, let's move forward. Right, so we have to talk a little bit more about primary and secondary growth. So as a reminder, remember, primary growth occurs in all plants, and its role is to make the plant grow longer, right? Increase in length. I'm doing this, length. Secondary growth only occurs in some plants, and the job of secondary growth is to increase in width. Because if you want to get super, super tall, you got to get a little thick. You got to get a little thick, because if you're really super skinny, you can't get to be 100 and something feet tall. You're just going to fall over. So you need to get thicker. That's right. The big plants get thick, and that's good. So all plants have primary growth. That's what I just said. some plants have secondary growth, the ones that want to get super, super tall. So let's talk about that a little bit. Herbs. So herbs are, or sometimes you'll see the word herbaceous, right? Earlier I mentioned grass, right? Grass is herbaceous, right? And so herbs by definition only have primary growth. Herbs don't have secondary growth. Herbs never get woody, right? Like a tree trunk, right? And this is because herbs don't need to get to be 100 feet tall. Most herbs, like the grasses I mentioned before, they don't need to get super, super tall. They don't need to make secondary growth. They're fine with just primary growth, so just stick with that, right? Why waste energy making secondary growth if you don't have to? So herbs never have secondary growth, only primary growth. Shrubs, on the other hand, have both primary and secondary growth. So, and that's because shrubs can produce wood. So maybe you're noticing a trend here, right? Herbs never have secondary growth. Herbs never have wood. Shrubs do have secondary growth, and shrubs can make wood, right? And then trees also are capable of doing secondary growth and also make wood. So that should link something with you that if something is making secondary growth, it will have a party. Wood. Thank you. Oh boy. Thank you for humoring me. Yes. So if something has secondary growth, it will ultimately make wood. Okay. All right. So hold on to your hats. We're gonna make it through this. It's gonna be okay. We already talked in detail about this primary growth stuff. We already talked about it. We looked at it under the microscope, right? We saw the cross sections, and we talked about how in primary growth you have that terminal bud, right, or apical bud. That's where the meristem is, the apical meristem. Apical meristem is going to divide into those 3 primary meristems to give rise to those primary tissues that we already have talked about a bunch of times, right? Epidermis, pith, cortex, xylem, phloem, right? A bunch of times now. This is a schematic that just organizes that exact same information. So remember, in purple is those primary meristems, and this is showing you which tissue each of the primary meristems gives rise to, right? Okay. Ground meristem, for your purposes, just think pith and cortex. You don't have to worry about these cell types for right now. Just pith and cortex. Okay. So that's primary growth. We already talked about that. It's primary growth. But now we need to get thick, and so we need secondary growth. And in order to have secondary growth, we're going to use lateral meristems. So primary growth, which job is to lengthen, uses a apical meristem at the tip to get longer. Secondary growth is for widening, and so you need meristems that go laterally out to get thicker, outside. Not outside, but that way, not that way, right? Lateral. And there are 2 lateral meristems. Here they're color-coded into yellow. And the names of those 2 lateral meristems are the Vascular Cambium and the Cork Cambium. So those are the 2 lateral meristems that are going to make the plant get wider. The Vascular Cambium, so remember, these were the primary meristems that we've already talked about, and they all created these primary tissues, right? Right? For example, Procambium made your primary xylem and primary phloem. Well, Vascular Cambium is going to make secondary xylem and secondary phloem. We're going to go through it. Don't worry. We're going to go through it. But the point is, I want you to take away their secondary growth. And in secondary growth, you have to get wider. And in order to get wider, you need lateral meristems. And in plants that do secondary growth, those lateral meristems are called Vascular Cambium and Cork Cambium. And we'll go through the tissue types as we move forward. But Vascular Cambium, Cork Cambium. All right. This sums up all of that on one slide. So remember purple is those primary meristems, right? And yellow is those lateral meristems, right? So here you go. You have the apical meristem here. Let's do this. Right? You have the apical meristem that's at the tippy tip. Apical meristem is going to divide into those 3 primary meristems, protoderm, ground meristem, and procambium. And then those are going to make all those primary tissues that we just talked about, these. And then eventually, don't worry about the transition right now, but eventually, right, you're going to end up with 2 lateral meristems that are going to give rise to your secondary tissues, okay? I like color coding, it helps me. So look, let's go here. This is just highlighting the meristems, primary meristems versus those secondary meristems. Don't worry about this. Sometimes students ask me like, well, how do we get those secondary meristems those lateral meristems? And you don't need to know this, but just FYI, they actually are created by some of those primary tissues, but you don't need to know that. Oh, I highlight this because sometimes you'll see this term paraderm. Sometimes you'll see this term paraderm. Your lab uses the word paraderm. Paraderm is a word that means all of these tissues together. Just FYI. So if you see the word paraderm, don't freak out. It just means all of these. It's referring to all of those things together. All right, too many words. Let's move on. Okay. So now I'm gonna walk you through a plant that is originally having primary growth and it's going to transition into secondary growth. So I'm going to walk you through this right now. So here is our plant, and there should be some familiar terms at this point in that we've been talking about this, right? So I will highlight it, right? Here's that apical meristem, right at the tip. We've already talked about. And so let's walk through. So that apical meristem is going to do the things we already talked about, right? It's going to create those create the primary meristems that lead to these primary tissues. So at the top of that stem, you have the top of that stem is still trying to lengthen, right? Still trying to get longer. And so it's having primary growth occur so that it can grow longer, right? Okay. However, down at the base of the plant, so right down here, and I'll use yellow, right down here, it's already starting to get thicker, right? Because it knows it wants to get super, super tall. So it already has to get a little thicker. And so if we were to look at that, right, I'm going to pull it out over here. Notice these. Notice the Vascular Cambium and notice the Cork Cambium, right? Oh, sorry, I'm so bad at that. So down there, there's already this transition happening. Now we already have lateral meristems down there that are going to start to give rise to these secondary tissues. So let's look at that. So this is a slide we saw from before, right? The primary meristems. And those primary meristems are going to give rise. So this is at the tippy tip, right? The primary meristem is going to give rise to those primary tissues. And then this is what's happening at that base of that plant, right? I'll use gold again because that's the color I used with this. You have this Vascular Cambium. And you have, well, they don't have the Cork Cambium on here yet. But the Vascular Cambium is going to give rise to, right, this secondary Xylem and secondary Phloem. Notice that you still have some primary tissues here, right? Like you still have Pith, right? You still have primary Phloem. You still have Cortex. You still have primary Xylem. And you still have Epidermis. That's because if you're going from primary growth to secondary growth, it's not like I'm primary growth, I'm secondary growth. Like it's more, if you think about it more, it's a transition. So I like to give the analogy of like a tadpole to a frog. So if you have a tadpole, it's not like you close your eyes and then it's a frog, right? Like first the tadpole might like grow little legs, even though it still has a tail, and then it might like grow little arms, right? And so depending on when you look at the tadpole might have a combination of tadpole-like qualities and frog-like qualities, but ultimately it'll end up being a frog, right? And so that's, thank you. I think it is a good analogy because it's the same thing is happening. This is a transition. It's not, you don't go primary growth, secondary growth. It's like you have primary growth. Oh, now I need to start transitioning to secondary growth. And at some, depending on when you cut the plant down and are looking at it, you can see both types of growth happening. But eventually, like the frog, eventually it's going to lose its tail. We're not going to have any primary tissues left. It's going to be a frog. It's going to be a frog or all secondary growth. So here, this one down here is almost completely just secondary growth, almost completely. So for example, let me get the blue. You can look at the color coding from the one above here, but right, so this secondary xylem is all this red stuff, and the secondary phloem is all this blue stuff, right? And now look at the outside of the stem. We no longer have just this simple epidermis that we had in primary growth. Now we're starting to see those tissues that the Cork Cambium makes toward the outside as the plant gets thicker. Let's look at it as a real thing. Let's not look at it as a diagram. Right. So, but the point of this slide is that the point of these couple slides is that all plants have primary growth. Some plants have secondary growth. In plants that are going to create secondary growth, they will have to transition from having primary meristems and primary growth to secondary or lateral meristems and secondary tissues. That's the takeaway. And eventually, like, for example, if you cut down a really old tree and you were looking at the stump, eventually, if you were looking at that stump, you're only going to see those secondary tissues because the primary tissues are gone, right? And now it's a frog, right? Now it's a frog. All right, let's move on to like looking at it in real. Oh, right. Ooh, this is fun. Okay. So I have, sorry, I get excited. I have some slides here of plants, and you have to tell me with looking with your eyes, right? I don't know what else you would look with, but looking with your eyes, you're going to tell me if you think that the plant has primary growth or secondary growth, right? So the point is, if it has secondary growth, it obviously also did primary growth, but you're going to tell me, all right, there's a question here, hold on. So are plants conducting secondary growth only developing ruptured epidermis? No, the idea is that, here, that's a good question. Let's go back. The idea is this. Yeah, I guess I kind of glossed over this a little bit. So look here. Let me get yellow because that's what color I used for the lateral meristems. Right. Here is one of the lateral meristems, right? Cork Cambium. Here's the other lateral meristem, Vascular Cambium. Vascular Cambium is going to give rise to your secondary phloem and your secondary xylem. That's in the inside of the stem, right? Secondary xylem, phloem, secondary xylem. However, the Cork Cambium is going to give rise to cork on the outside of the stem and Phloem on the inside of the stem. And so what that means is ultimately, once you are in full secondary growth, you won't have epidermis anymore. Epidermis will be ruptured. The epidermis is going to rupture, and in its place is going to be cork on the outside of the stem, and just beneath the cork, the Phloem. So if you look at this here, hey, there we go. Right. So what's happening in this bottom slide is that the epidermis is being ruptured because it's not going to be needed anymore. Bye-bye epidermis. Because now we have secondary growth. And so you see this brown stuff here? That's your cork and Phloem. So the cork will be toward the outside of the stem and the Phloem be toward the inside of the stem. As we move forward, we're going to see some other slides that should make this a little bit more clear about what those parts are. All right. Oh, no. Okay, so before we do that, let's play this game. So I'm going to show you a plant, and you tell me if that plant has primary growth or if it's capable of making secondary growth. All right. So here's the first one. Got a little basil plant here, right? Basil, which is the species name, is Ocimum basilicum, right? That's why we call it basil, is basilicum. So here's a basil plant. What do you guys think? I have a few, right? So everyone is saying primary growth. Correct, right? Correct. So let's go to the next one. Now we have a lemon tree, right? Which is Citrus limon, which is why we call it lemon tree. Okay, great. So I'm seeing a lot of people saying secondary. I'm saying some people are saying both, and or capable of secondary, and absolutely correct. Absolutely, right? If you're not, one key is this, right? So that's where we're starting to see thickening. But yes, there's probably primary growth occurring up here as it lengthens. Very good. All right, next. All right, so this is a plant called crown of thorns, or Euphorbia millii. What do you think? What do you guys think? So this plant is, all right, I got a lot of, all right, so people are saying primary, some people are saying secondary, right? This one is tricky. So I think in this plant, well, I know, but this one is a little trickier. So there's definitely primary growth occurring up here, but the stems are slightly woody, right? They're thick and hard, slightly woody, and so I think both are happening here. But it is a trickier one. This one is definitely trickier. But if you think of this, right? This is similar, right? Okay, in that you have both happening, but this down here is kind of the clue. Okay. Ficus tree, right? Ficus benjamina. So which type of growth is this plant? What types of growth, what type of growth is this plant capable of doing, right? Yeah, so everyone's saying secondary. That's good. Yeah, so again, right, you have down here, we can see the woody structures, right? We can see the cork on the outside of the bark. And then up here, there's probably primary tissue, primary growth occurring at the tips. Good. All right, I think this is our last one. Yep, so this is corn plant, very common house plant. Okay, cool. So I have a few people responding to this corn plant, and everyone is correct in that, yes, it's also capable of both, right? So up here, you are probably having you're having primary growth occur. You probably are also having primary growth occur even like right here, where these little guys are lengthening. Yeah, someone says I have one of those in my living room. It's gorgeous. See, the very common houseplant. It's part of the reason I picked it for today, because I think most people have probably seen this in one time. Yeah, but the woody structure, and you can see it's already getting thicker, right? Is your example of the secondary growth, and so yes, capable of both. Okay, great job, guys. Okay, how much more do we have? All right, we're getting there. We're getting there. Do you guys want to take like a 5-minute break, or something like that? When do you take those? No, people are saying no. You guys just want to finish. But some people are saying yes. So we have 2 no's and 1 yes. Oh, that's not, all right. We got 2 no's, 3 no's. Many, okay. No breaks. All right, fine. We'll truck along. So yes, you're overruled. Sorry. All right. So let's talk twigs. Let's talk twigs. So there was a question, when do you repot? I think it was when do you, sorry, I missed it. It went away.
I just wanted to know, when do you like take the plants out the pot? Like
for these guys?
Right.
So in general, these plants do like to be pretty snug. And so if anything, I mean, I've seen them when they're literally like breaking the pot. Like if it's in a plastic pot, they're still doing fine and they're starting to like rupture the plastic pot. The ones that are in this picture here, I would probably pot them into a pot that's like just a little bit bigger than this. But you don't want to take it from something like, here, I'll draw. You don't want to take it from something that's in this size and then transfer it into a pot that's like, you know, like this big. Because the root is only this big. And so what happens if you put a plant here, let's draw a pot. If you put a plant with a root that's only this big here into a really big pot, what'll happen is here's the root, it'll be just at the top. And then when you add water, the water will go and it'll settle at the bottom of the pot, but the roots can't even reach it. And so you can end up with root rot. See, they can't even reach it. If you feel like it's been a while and you want to repot it, I would repot it, but only repot it to like the next size. People a lot of times think if you put a plant into a bigger pot, it'll somehow make it bigger, like a fish. But I always say that a plant, it's more like your foot. So if you wear a size 6 shoe, just because you put it in a size 13 shoe doesn't mean your foot's going to get bigger, right? You want to wear the right shoe for the right size of your foot, and the plants are very much like that too. Okay, let's talk twigs. So a twig is like a little stick, right? That's on plants that get woody. And let's go forward. I have my thing here. So first, this is something that the lab covers. We're going to talk parts of a twig. So some of these things should look familiar, right? Some of these things should look familiar already. We got our apical bud right at the top, right? We have our lateral buds, which are capable of making branches. And then now we can talk nodes and internodes because these are parts of a twig. So a node is a region, a location. An internode is a region or a location. At the region of a node, so here's a node, you have axillary buds. So the axillary buds occur at the node. Again, a node is a region. And then the area between nodes is the internode. Inter means between. So the word literally says between nodes. So if this is a node here, and this is a node here, etc., this is an internode. This is an internode, etc. Internode. So let me move. So also around the node, depending on certain things, I want to draw your attention to this here, bud scale scar. Bud scale scar. So here, this is that apical bud, right? This is that apical bud. And the apical bud, it's almost like a little, what's the best way to think about it? It's almost like a little hat. The apical bud is almost like a little hat on the very top of the stem, twig. And when the apical bud starts to grow, so I don't know if you walked around in springtime, you may have seen plants with buds on them, and you were like, "Ooh, the leaves are coming soon, right? It's gonna be leaves soon. I could see buds on the trees." Or maybe you don't even look at trees, and so you're like, "What's she talking about?" But maybe you did. And what happens is when the plant is gonna undergo primary growth, the first thing that happens is that the bud scale, the little hat, falls off. And when it falls off, it leaves a scar. So for example, let me pull up pink. So you see this little, it almost looks like a, I don't know. It looks to me almost like a Pokemon type of thing, but I don't know. But the thing at the very top of the twig, that whole thing is a bud scale. And when the twig starts to undergo primary growth, that bud scale is going to fall off. And when it does, it's going to leave a scar. So it's going to leave a scar right there. And that scar is going to go all the way around the twig because it was on the whole surrounding the top. And each year, so look, here is an old bud scale scar from when that bud scale scar, when that bud scale fell off. And then look at how much growth the plant did in that year. And so the point of this is that each year, where's pink? There will be one bud scale scar per year. So you can tell how old a twig is by counting the bud scale scars. Here you go. Here are some twigs, right? And every year that bud scale is going to fall off, right? So the little hat, here's the little hat. Here's the little hat. Here's the little hat here. Each year when active growth is going to start happening, the first thing that's going to happen, that's going to fall off. And when that falls off, that's going to leave a scar. It's going to go all the way around the twig. Here's another one. Here's another one from previous years. Here's another one. Why is this helpful? So this could be helpful for if you're trying to pick landscape plants for your yard, or maybe you were given a plant by a family member and you're trying to figure out how old it is. This is one way you can actually tell how old parts of that plant are. Is you can tell, like for example, the twig on the bottom here is 3 years old. The twig on the top here, oh, you have to remember, right? We're not seeing the whole twig, right? So there could be more bud scale scars going this way that we can't see. But the point is that you can learn a lot about this. Like you can tell how old that part of the plant is. In addition, you can tell about growing conditions. So for example, look how much, oh, that's not what I want to do. Look how much this twig grew in 1 year as versus look at this year. Well, let's look at a finished year. Look at this year, like half as much growth. So it can tell you about whether or not the actual plant is happy as well, right? If you're seeing stunted growth over years, you might want to, like you said, repot it into a bigger pot or maybe start fertilizing or things like that. So that's why I'm teaching you how to age a twig. Okay. This is just an empty twig slide for you to have in case you wanted to practice like this thing, but this is the twig that they have in the lab manual. So looking at this twig, how old is this twig? Oops, go back. Hey, go back. If you were looking at this twig, how old is this twig? So we have bud scale scar, we have bud scale here on the top. This is going to fall off. So there's that. But looking at the rest of the twig, right? Do we see any of those lines, right? Those bud scale scars. How many do we see? Everyone took a break, even though I didn't.
2?
Yeah, thank you. Yes, it's 2. Here's one. Here's another. So this right here is 1 year's growth. This right here is 1 year's growth. We can assume there's at least another year below here. So we would say this twig is at least 3 years old, right? At least 3 years old. Thank you. Thank you. All right, let's move on. All right, we got to talk a little bit more about that secondary growth, and then we're gonna talk about secondary growth in mature stems, right? Those thick stems. We're gonna talk about variations on stems, and then we're done, right? So maybe 15 minutes. All right, mature trunks. Here is a mature trunk. So remember what I said, you can't get tall unless you get thick. And look at this tree, right? This tree is very, very tall, and look how thick it's gotten, so it can be strong enough. So we would look at this tree and see that this tree has abundant secondary growth. Oops, abundant secondary growth, right? It's very, very thick. And recall that we already talked about this, right? In secondary growth, you have those 2 lateral meristems, the Vascular Cambium and the Cork Cambium. The Vascular Cambium is going to give rise to secondary xylem, secondary phloem. And we call what you and me call wood, like, oh, I have hardwood floors, or look, I need a new wooden door, or I have a boat and it's made out of wood, or a bat. Here's a baseball bat. It's made out of wood. What you and I call wood is actually secondary xylem. secondary xylem, aka wood. So if you really want to nerd out, you can be like, I have secondary xylem wood. I have hard secondary xylem floors, or I need a new secondary xylem door. Wood is secondary xylem. What you and I call wood is secondary xylem. So Vascular Cambium gives rise to secondary xylem, secondary phloem, and Cork Cambium gives rise to cork on the outside of the tree, and then Phloem toward the inside. Let's look at it. Oh, no annotating. So here is what we call in the business a tree cookie, right? This is a slice through a mature trunk. And you can actually observe these using a slice of a trunk. You could actually observe these secondary structures that we're going to talk about. So let's first look at a diagram, and then we'll look at it under the microscope. So here's my trunk, right? And now I'm going to look at a diagram of these tissues. So let's use my same as before. Let's use the yellow. Yellow is what we're using for the, so notice here is one of those, oh gosh, man, I'm so bad at that. I need like a stylus or something. All right, so here's our Vascular Cambium, right? So this is a lateral meristem, and it's going to be right there. Here's our Cork Cambium. This is our other lateral meristem, and that is right here. Vascular Cambium is going to produce secondary phloem toward the outside of the stem, and Vascular Cambium is going to produce secondary xylem toward the inside of the stem. Remember, secondary xylem is what we call wood. Cork Cambium, lateral meristem, is going to produce cork toward the outside of the stem, and Phloem toward the inside of the stem. Okay. Well, that will be clearer on these next things here. So there's those lateral meristems highlighted. Let's first talk about the Cork Cambium. This is what I just said. Cork Cambium produces the cork toward the outside of the Cork Cambium. You can see it here in blue. And then Phloem, which is another type of tissue that grows toward the inside of the trunk on the inside of the Cork Cambium, right? And then, as I mentioned in that earlier slide, all three tissues together, you can refer to them as the Protoderm, but that's just a term that shows up in your lab. This is the second lateral meristem, right? The Vascular Cambium. Vascular Cambium is producing that secondary phloem toward the outside of the Vascular Cambium, right? Toward the outside of the stem, trunk. And then the secondary xylem, aka wood, is going to grow toward the inside of the Vascular Cambium. Okay. So remember when I was showing you those slides before, eventually, if you're looking at really mature stems, eventually all of the tissue inside of that stem is going to be secondary tissue, right? So to kind of recall back from before, this trunk that I'm touching, that's actually in Queens and Alley Pond Park, this trunk doesn't have epidermis anymore. This trunk doesn't have pith. This trunk doesn't have cortex. This trunk doesn't have primary xylem or primary phloem anymore. This trunk is mature and only has secondary tissues present. And the majority of this trunk is composed of wood, aka secondary xylem. So pretty much this whole inside is just secondary xylem. And the other tissues make up very little of the trunk. All right. So just like I showed you with twigs, where you can age twigs, you can age trees. You can tell how old a tree is if it's cut down. So I'm showing here, secondary xylem, aka wood, is abundant, right? Like in this example. And this tree, by the way, I counted, and it's about 169 years old, by the way, when they cut that tree down. The way that you can tell how old the plant is or was when it was cut down is because the secondary xylem produces, there's a ring of secondary xylem every single year. So every single year that the plant is alive and has secondary growth, you're going to have a ring of secondary xylem each year. Don't worry about tracheid cells. It's a ring of secondary xylem each year. And you'll be able to see wood rays, which are just other structures. But my main takeaway from here is that one ring of secondary xylem every year. Let's look at it. Oops. Okay, so this is a close-up. Here, if we go back, see here, secondary xylem tissue, this area of this diagram, that's just a close-up of that here. So look, secondary xylem is abundant, right? 1 annual ring per year. And what it looks like under, if you're looking at it closely, is there will be, so trees grow really fast in the spring when there's a lot of water and the temperatures are pretty cool, but then they grow slower in the summer because there's less water and it's super hot. So each year you'll see 2 types of cells. You'll see this type of cell. See how those are round, like see how those cells are all big and round? That's what we call early or spring wood. So this is secondary xylem that would have occurred, that would have been grown in the spring. And then this, where the circles are a little tighter, those are late or summer wood. And such that each year, this is 1 year, right? Spring wood, summer wood, end of the year. And then this tree, when it was cut down, this is all spring wood. We don't see the summer wood yet. So let's look at it on an actual tree here. So look, you might have heard of people talking about tree rings before, right? So each of these rings is 1 year of growth that this tree had after it reached the secondary growth phase. In the case of this tree, it's a little hard to see the early and spring wood, but the early wood is probably darker in color, and then, or sorry, the early wood in this case is the lighter color, and then the darker wood in this case is the late wood. But you could see if here, if you just count the lighter colors, you could see a ring, right? So 1, 2, those are all a year of growth, right? Sometimes toward the outside, it could be a little hard. Sometimes toward the very, very center, it could be hard to tell the numbers. But like I said, yeah, that tree that I showed myself with, that was 169 years old when it got cut down. So it was probably about 175 years old because it would have had primary growth before secondary growth. So annual rings is how you can tell how old the trunk is. But then you also have these things that are called wood rays that radiate out from the center. So it's not super visible in the previous, but here they're more visible. So let me draw them here. So you see all these lines kind of radiate like a bicycle wheel spoke, right? Like a spoke of a bicycle wheel. These are made up of special type of cells that allow food to be vectored from the inside to the outside. But the point is, so annual rings are able to age the tree, and then you'll also see these wood rays. And don't worry too much about wood rays, but that's what those lines are. Okay. So we looked at it in diagrams. We looked at it in a tree cookie. Let's look at it under the microscope. So this is a, and then we're almost done. So this is a stem that has secondary growth, and we're going to look at it under the microscope. Let's see if there's anything here. Yeah, so don't worry about here. Don't worry about resin canal. So look, this tree still has some cortex left, right? So this tree still has some of those primary tissues left. The reason is because this had to fit on a microscope slide, right? A microscope slide is this big. So there's no way that we could have done a microscope slide that would have all secondary growth, because the tree would have been like, you know, that small to be able to fit on the microscope slide. But the majority of the things that we see here are those secondary growth tissues, right? I'll keep using black because the thing here is primarily blue. But look, so here, vascular cambium. So here's your vascular cambium right here. On the outside, toward the outside of the stem, is where the secondary phloem is growing. And then toward the inside of the stem is where you have your secondary xylem, right? AKA wood. You can see the annual rings. So here's one annual ring. Here's another. Oh, yeah, I'm really bad at that. Oh, that's not working. Point is, yeah, so you can see all the annual rings. You can see the early wood and the late wood, or the spring and summer wood, right? Look at here, spring wood. See how big the cell structures are? Late wood, summer wood. See how condensed they are? So each of those will be present each year. And if you were to age this tree, you would say that this tree is in its 4th year of being alive. All right. Enough about stems. Enough about the inside of stems. It's like I said, it's not the most exciting material, but this part's fun. This part's fun, and we're almost done. We got like maybe 5 minutes or so. So we saw stems, primary growth, secondary growth. Now let's look at what are some adaptations that stems have undergone over evolutionary time to do certain different functions. 1 second. Sorry. Okay. Let's do this. All right, so let's look at these. So one type of variation of the stem is what's called a liana or a vine. So you may have seen these before. Liana just means a woody vine. So a liana is a vine that's capable of making secondary growth, like you see here on the left. And then vine. And the vines just allow plants to climb. Next, you have tendrils. Tendrils are these little curly-Q things. So these are modifications of the stem to allow plants to do different functions. Tendrils also allow plants to climb. So you can see here the tendrils are wrapping around this stick to be able to allow the plant to grow up. Another adaptation that some plants have is thorns. So the crown of thorns that we looked at earlier has thorns all on the stems. And here in a black locust, also you can see those are some pretty vicious stems. And as you can imagine, stems are to serve for protection, so that the plant is protected from predators. Another variation on the stem is what's called rhizomes and stolens. And what these do is they allow a plant to spread to new areas by creating basically clones of itself. So let's use black. So look, this is a new plant. This is a new plant that this main plant is creating by using these special stems called rhizomes and stolens. Rhizomes and stolens are basically the same thing. The only difference is a rhizome goes underground and a stolon goes along the surface of the ground. And so this will continue growing, and a new plant. Sometimes if you've weeded your garden, sometimes you might have pulled up what you thought was one plant and then like 10 plants come up because they're all attached by these rhizomes and stolens. Some of our weedier plants have a tendency to do this, which makes them more successful as weeds. Okay, and then last but not least, we have the corm and the tuber. The corm and tuber are examples of stems that store food. So for example, a potato is actually a type of a stem, believe it or not. So a potato is a stem. Ultimately, roots will grow off of it. Right, here's some roots, they'll grow off of it eventually. But a potato is a modified stem. Some of your favorite spring flowers, like here I show you on the left, crocus, grow from corms. And those are also examples of a modified stem that stores food. Okay. Whew, stems. Not the most exciting, but important to understand. So that sums up everything for stems. We do have a homework assignment on stems that's available it'll be available at starting at 8 p.m. on Blackboard and is due anytime between now and Saturday by 6 p.m. I will check all of the assignments that you guys submitted for the first assignment. I will check them, make sure that there's no errors. And then I will, after the due date, which was today, I will post, I'll change the settings so that you can see what answers you got right and wrong. So in the future, the answers to the assignments will be posted after the due date. Cool. Yeah, and that's it for stems. Next class we have the leaf. I'm pretty sure that next class I'm going to be posting a recording. I'm not 100% sure yet, but I think I am. What we'll do with that is, and I'll put instructions in Blackboard about how to do that. So if I post a recording of this, I'm going to set it up in Blackboard where there's a discussion place on Blackboard. And so when you're watching the recording, if you have any questions, I want you to write it in the discussion board on Blackboard, and then I will go to the discussion board and answer all the questions so that everybody can see the questions that are asked. Okay, but I will send you information about that if we do a recorded lecture or lab on next Tuesday. Okay, so yeah, if you guys have no questions, thank you very much, and I'll post this recording once it finishes processing. The lecture slides will also, I think, be available starting at 8. So thank you guys very, very much. Thank you guys. Oh yeah, have a great weekend. Yep, sorry, I was behind on the chat box. Thank you guys. Have a great weekend. Stay safe.